Okay, hi everyone. My name is Isabel, and along with myself, Rachel, and Drew, we are the StaRS Filtration Theory subteam. So STAR stands for Stacked Wrap Sand Filtration, and it is crucial to the water treatment process as one of the last steps in Agua Clara's water treatment. STARS filters are responsible for removing the last of the unwanted particles. In order to operate effectively under the Environmental Protection Agency standards, these filters must reduce the turbidity, which is essentially the degree to which water loses its transparency, to about 0.3 NTU or less. And efforts to better refine these filters, variables such as grain size, the height of the active zone, which is where the clogged and empty pores meet, and coagulant dosage will be tested in order to increase the failure time, which is the time that it takes the system before it has to be backwashed and essentially restarted. At the beginning of experimentation, the fall 2019 sub-team focused specifically on the grain size. This semester, we weren't able to go into the lab, but this is something that we will look into in next semesters. So as I said earlier, STAR stands for Stacked Rapid Sand Filter. And essentially these filters are used, they use sand to catch particles in the water. And then in doing so, it filters the water in the process. This filtration is one of the last steps in the 4-phase process for water purification. And essentially STARS is the step right before water purification, and it prepares the water for those processes. A STARS filter consists of water pipes sandwiched between layers of sand so that water can flow in and out of the filter freely. The entire filter is generally housed in a PVC casing inside a concrete or cement enclosure. So how it works, here we have some filters basically to demonstrate on a very general level how the filters work. And so when water enters the filter, it has already been mixed with a coagulating agent to create what we call flocks. And these are large groups of undesired particles in the water bound together by coagulant. These flock particles get trapped in the filter by colliding with sand grains and adhering to them. So the dirty water coming in emerges as filtered water, which is ready to be purified in the next steps of the process. In the left picture, you can see kind of this in action. The tan spheres represent sand grains and the black particles represent flocks, which are these undesirable particles that are bound together by the coagulant. And then on the right-hand side, you can see just kind of a figure that demonstrates that there's dirty water coming in, then it flows through these stacked sand filters and it comes out as filtered water.
So in a nutshell, a basic sand filter, as discussed before, has dirty water coming in, which then gets purified through the sand filtration process. The STARS model has multiple filters stacked above one another, as pictured on the right, and essentially figure 4 on the left is just one unit of many in figure 5. As the water goes through the pores of the sand and reaches the bottom of the filter, it exits through an outlet, and in the case of figure 5, there are multiple outlets. Because space is sometimes at a premium, multiple filters are used to increase the efficiency of the entire setup by decreasing the amount of space required by using a series of filters stacked upon one another, as you can see in figure 5. Next slide, please. So the last main step of the filtration process is the backwash, where the clean water travels at a high flow rate up through the sand filter. This essentially scrubs the sand grains of the flock particles so that when water flows in reverse, flock particles are removed from the filter, and this wastewater is further processed into sludge. Once the backwash is complete, the filter is ready to be used again. Now, this is the crux of STAR filter theory's research. We're striving to increase the time that the filter can perform its function effectively before needing to be backwashed, which Rachel will be elaborating on in a little bit. But for now, next slide. The issues that sand filters have in terms of performance are about clogging filters and are about clogging the pores in the filter. After a while, flock particles will occupy a significant amount of surface area within the filter, making the cross-sectional area of the pore smaller. This is a problem because the flow rate of the water is going to be constant. So that means a smaller cross-sectional area will lead to a higher water velocity. So when the water starts to flow faster through the pores, it makes it harder and harder for flock particles to adhere to the sand particles and be filtered out of the dirty water. So this just results in dirty water coming out of the filter, which is when the filter must be backwashed.
So our main focus is going to test the sand grain size, as we have mentioned. And so the team from the 2019 and 2020 StaRS Filtration team did work on testing the grain sizes. However, there was many setbacks due to simplifying the model and updating the equipment. And the reason why we're testing sand grain size is because there's a study that noted that the larger the grain size, the faster the wastewater flowed through the sand, and a greater amount of wastewater can be filtered. And an effective place to start was noted in a study from the Ohio State University that an ideal sand size would be around 0.3 millimeters to 0.5 millimeters, which is a helpful starting place for our team when we get into the lab. So as I mentioned, the 2019 team made a more simplified model of the filter where you notice that on the left, they used to have 3 filters and 4 turbimeters. And however, they made it more simplified to have 2 turbimeters and 1 filter. This way, it allows for water to enter through the filter and straight through the backwash, so there won't be any unneeded cycling of backwash water. So this is what the lab apparatus looks like from the 2019 team. However, we did visit the lab once, and we noticed that it no longer looks like this. So once we get into the lab, I'll mention more later how we will kind of have to start from scratch and rebuild the apparatus again. Okay, so since we mentioned that we have not gone to the lab in person, we just had to prepare for it by looking mostly through the Python code and the Procoda method files. And so the Python code we found was modified and was used to calculate the coagulant dosages for the setup, as well as the flow rates for each pump in the setup. The Procoda tutorial helped us set up a customized method file for running experiments in the lab. In the next slide, we'll go over a few relevant set points, which are essentially the cogs in the Procoda operation. For instance, we'll set up the run and warm-up states, which lets us set up time conditions for experiments and /test runs.
So we spent a majority of the semester looking at the Python code from previous semesters and trying to make sense of it. And shown on this slide is a little snippet of the code. And so this part of the code essentially generates the water flow rate as well as the volume of water per revolution and the water pump RPM, which can be seen by the print statements. And the code, essentially the rest of the code also just uses formulas to generate these types of constants and flow rates and such that can be used in the rest of our experiment and also can be inputted into the Procoda that we set up. And while looking through the code, one major point of contention was the coagulant dosage range. So in this code, you can actually see it right at the bottom. It says that in a comment, it says the coagulant dose range is 0.5 to 8 milligrams per liter. But then in the other code that we looked at in previous lab reports, the group seemed to be using 0.2 milligrams per liter. So next semester, something we're going to look into is the optimal coagulant dosage that we should use in our experiments.
Yeah, so this table continues for quite a few rows below the slide, but just to illustrate what the set points we're using in our method file are, please direct your attention to the light blue arrows in the middle of the screen. The water pump ID set point allows Procoda to connect with the appropriate pump model and therefore collect data from the specific pump model accurately. Combining set points such as like the water volume per rev and with the water pump ID allows us to automatically calculate the flow of water from a particular pump in real time. Another example are the dark blue arrows. The set points plant flow and water flow are related in that the plant flow is a constant, 0.5067 milliliters per second, but the water flow can be calculated as a function of the plant flow minus the coagulant flow. And so this relationship shows how Procoda uses set points.
So our future tasks, so once we get in the lab, we will start setting up the filter, and we're going to run simple experiments to make sure that everything is running smoothly. And then after that, we can actually run our Procoda method file that we worked on and the Python file to make sure we have the right values for the flow rates and the dosages. And as we said, our ultimate goal is to increase the filter failure time, which expands on the previous year's works. And if we have more time, we will test the variables of the height of the active zone and the concentration of the coagulant. And if you have any questions or recommendations, feel free to email us here.